y'all, it's Mr. Reardon once again. It is still Monday, October the 27th, and I am still talking about cell respiration. Again, I want to give you guys a solid review so that you are confident in understanding these so fundamental biological concepts. And now, like always, we are brought to you by a beverage. Today, or our last video of the day, is brought to you by Diet Coke. Diet Coke, the sound of success. Okay, let's be real clear. Is anyone ever going to put a gun to your head and ask you to tell them the steps of cellular respiration? No. Is anyone ever going to offer you $1 million if you can correctly describe the fate of glucose within cells, beginning with glucose and ending with ATP and water and carbon dioxide? Highly unlikely. However, if you understand these fundamental biological concepts, you will have a better understanding of how your cells operate, and you will know more than most people know, and that is cool. If you've been around me enough, you know that I love this stuff. And most of my colleagues in the AP Bio world, IB Bio world, know me as the mitochondria guy. I mean, you know, all organelles are created equal. Some are more equal than others. The mitochondrion is one of those organelles. And this, not only is it just super cool that this is happening in all your cells all the time, but really, if you get down and study this stuff and thrash with it and walk away with an understanding of how your body uses the energy that you take in through eating, you really can probably figure out anything within the scope and sequence of this IB biology course. So I say all that with encouragement, but I also say that you can't just watch this and go, yeah, I got it, and walk away. You won't get it unless you're just a super genius. You have to get down and work with this stuff. And we begin with a figure from your textbook. This is figure 915. This is kind of the end, or sort of the back two-thirds of the cell respiration chapter. And what we're looking at is a fairly complicated diagram of the inner membrane of the mitochondria and then the embedded proteins of the electron transport chain. They're also showing you the flow of electrons and trying to show you the flow of hydronium ions. And then they're also trying to show you how ATP synthase taps this proton gradient to make ATP. And this is a beautiful figure, and you need to look at it. You absolutely need to read the caption below it. You need to read this chapter. But I've done this for years, and I watched students struggle with this concept, and so what I did is I went ahead and made a couple of diagrams myself that will hopefully clean this up, and that's what we're going to look at during this screencast. We're going to focus specifically along the inner membrane of the mitochondrion and look at how energy conversions are coupled to make ATP. The first diagram I am not writing home about, but it is slightly helpful, and it is right here. So here we have some cross sections of mitochondria done by yours truly. Imagine somebody took a mitochondria and peeled back the top. What you're looking at is the double membrane, and then sort of trying to allude to the inner folds of the membrane, that is the cristae, increasing its surface area. And then you'll notice that you have replicant copies of the electron transport chain and ATPase or ATP synthase along the electron transport chain. Not just a dozen like we have here, but probably thousands, if not tens of thousands, of copies of the electron transport chain and ATP synthase within each mitochondrion. And then here I want you to imagine we took a mitochondrion shaped like a hot dog and cut it right down the middle, so you're looking at truly in cross-section. And here we have the folds of the cristae, and here we have three replicants of the electron transport chain along with their ATP synthase molecule associated with them. So, and I'm trying to suss that out a little bit. We need to realize the electron transport chain is one thing. It does something which then allows ATPase to function. That's important and worth repeating. The electron transport chain and ATP synthase, they work together, but essentially ATP synthase cannot work without an effective electron transport chain, and that is definitely worth knowing. Now, the last figure that we'll look at, and here's where we'll spend most of our time, is a diagram I built of figure 915. And so what we're looking at here, I want to make sure you know you're in the mitochondrion, but I've cleaned out all the color and tried to make things a bit more straightforward. When you look at these dashed lines right here, we're looking at what we call the inner membrane of the mitochondrion. And then, just like everything in your book, purple is proteins. Here we have PC1, 2, 3, and 4. That stands for protein complex 1, protein complex 2, protein complex 3, and protein complex 4. Your book talks about these 4 protein complexes. Worth noting that these protein complexes are actually the conglomeration or amalgamation of over 90 proteins working in concert to generate a proton gradient. Also embedded in the inner membrane is an organic molecule called ubiquitone, which we call Q, and then also what's known as the cytochrome C complex. Cytochrome C, a very important set of proteins and pigments that help strip electrons and move electrons down the electron transport chain. Also of note is this beautiful protein complex over here on the right in a deep royal purple. We call it ATP synthase. It's associated with these it's associated with the proteins of the electron transport chain. And then we're getting some more orientation. On the underside of this figure, we have the matrix of the mitochondrion, and then you'll notice this thin area here, the intermembrane space. This is the area between the inner membrane and outer membrane of the mitochondrion. This is going to be incredibly important in just a moment. Also of note, we've got our electron shuttles, both NADH, teal from earlier in our screencast, and then also FADH2, which is this kind of beautiful coffee color, also from an earlier screencast. And then none of this stuff happens without oxygen. So oxygen is present in the matrix of the mitochondrion. We breathe in, oxygen diffuses, is really pulled into our lungs as we expand our thoracic cavity, and that oxygen diffuses across the alveolar surface into our bloodstream and then is transported to our working cells all along a concentration gradient. Eventually that oxygen makes it into the matrix of our mitochondrion, and that's where it's going to be most essential. Now, something real quick that you need to know. What you're looking at here is a series of redox reactions, reductions, oxidations. When we talk about reduction, we are talking about the loss. I'm not going to screw this up, Miss Anderson, I promise. The loss of electrons is oxidation, and the gaining of electrons is reduction. And so that's kind of weird, right? You gain something and you are reduced. What that means is your electronegativity is reduced. You become more negative when you become reduced. So think of oxidation as a breakdown or a loss, and think of reduction as an increase in complexity. And you'll see that here. The thing you need to realize is that when we look at the protein complexes of what we call the electron transport chain, is that each of these neighbors is more electronegative than the neighbor up the stream. What I like to tell my students is that oxygen is a bully, and it picks on protein complex 4, and protein complex 4 is a bully and picks on cytochrome C, who's a bully, who picks on protein complex 3, who's a bully, who picks on protein complex 2, who picks on Q, who picks on protein complex 1. So we sort of have this gradient of electronegativity. We also have this, if you want to think about it in kind of a cheesy, schoolyard bully context, we have the gradient of bulliness, like protein complex 1 is a bully, but not really. Oxygen's the biggest bully on the block. And so keep that in mind. Here's the scoop. NADH is either being imported from glycolysis, or which is coming from outside the cell, or it's coming from the Krebs cycle. But the thing you need to realize is that NADH and FADH2, they have electrons, and they are total wimps. So let me change my colors here real quick, and let's follow the electrons down from FADH, or NADH all the way to protein complex 4. So what's going to happen is NADH is going to easily give up its electron, and those electrons are going to flow down the electron transport chain and ultimately get bound to oxygen. Secondarily, FADH2, it donates its electrons to protein complex 2. I'm going to try and got my colors right here. And FADH, those electrons are going to also flow down the electron transport chain to oxygen. And so we've got that continual flow of electrons from these electron shuttles to oxygen. And ladies and gentlemen, this is going to be constant as long as there is food to be oxidized and broken down, and as long as there is oxygen within the cells. These electrons are going to flow. For you guys that have been with me talking ecology, we always talk about heat leaving a trophic level. Well, as these electrons flow down the electron transport chain, they generate heat. And much of that heat is used to create an optimal temperature for enzymes, but also a lot of that heat is lost to the environment. So if you want to think about where heat comes from in an Eltonian pyramid, it's coming from this flow of electrons down this inner membrane, which is essentially an organic wire. The flow of electrons, just like electricity flowing down a copper wire, is happening here. Now, so what? So what? What good is all this electron flow? That's where the second piece of the puzzle comes in, and that's where things get really, really interesting. And so what I want you to imagine the dynamic flow of electrons across the electron transport chain, but then I want you to realize that as those electrons flow, we're going to get a movement of another set of ions from the matrix to the intermembrane space. Hopefully you guys can see that. So now we have 4 arrows popping up. As electrons flow, what we're going to notice now is that hydronium ions, H+, these guys are going to be pumped from the inner membrane of the mitochondria. Get those going here. These ions, they're initially in the matrix of the mitochondrion. As these electrons flow, that generates a charge. These proteins, there are certain proteins within these protein complexes that have the ability to pull these hydronium ions from the matrix to the intermembrane space. So as the electrons travel down the electron transport chain, hydronium ions, or protons as we call them in biology, are pushed against their concentration gradient into the intermembrane space. And that is a big, big deal. The flow of electrons, coupled with the active transport of hydronium ions, sets up what we call an electrochemical gradient. And so when you think about it, you can think about a charge across the inner membrane of the mitochondria similar to a battery, and what you're going to get is a positive, 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 positive gradient on one side of the membrane, and then on the other side of the membrane you're going to get a negative charge. And so you can see that here. You love it, you live it, you can't live without it. Negative, 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 negative. Here, check it out, man. We got a positive charge on one side, we got a negative charge on another. We call this an electrochemical gradient. Chemical, electric because it's positive and negative. Chemo because we've got hydronium ions trapped on one side. Now, you guys are smart. You pack all these hydronium ions next to each other. What do they want to do? They want to get the heck away from each other. They want to get out of that tight space. And so there's only one way those hydronium ions can get across the membrane in a normal functioning mitochondrion, and that is through ATP synthase. So this is the beauty of the system. As the electrons flow, they pump hydronium ions up against their concentration gradient. And then those hydronium ions flow through ATP synthase. That's basically a form of facilitated diffusion, you guys. And that's a spontaneous reaction, these hydronium ions flowing along their concentration gradient. That energy is used to convert adenosine diphosphate into everyone's favorite molecule in biology class, mine included. Trying to get this going here. It's going to convert the energy which is released as the hydronium ions flow down their concentration gradient is converted to the synthesis, if you will, of adenosine diphosphate and an inorganic phosphate into my favorite molecule, adenosine triphosphate. And you might ask yourself, what happens to the hydronium ions? Well, here we have a positively charged ion. Here we have an extremely negative molecule, this oxygen. It picked up all these extra electrons, and that O2 is simply converted into something as harmless as H2O, water. Super cool. Organisms that have mitochondria have the ability to take a very electronegative molecule, oxygen, a radical, a free radical, which can rip things apart and convert this dangerous atom into something as harmless as water. And when that happens, the cell is able to convert, the cell is able to turn, how do I say this best? The cell is able to make ATP or generate ATP from food. So I know that's a lot to take in. That was not my best lecture. It's easier with a whiteboard and a dry erase marker, but I think you get the point. The biggest point being, as electrons flow, hydronium ions are pumped against their concentration gradient. This sets up an electrochemical gradient along the inner membrane of the mitochondrion, and then that gradient is tapped by ATP synthase to make ATP. And without putting too fine a point on it, that is a great example of chemiosmosis. You need to watch this a couple times. You need to watch my other screencasts. You need to read your book. You need to ask me questions, and I will see you in class.